Shri Gurubhyo Namaha. Welcome to our online learning initiative and I welcome you all for the biology class. Last class we saw about trihybrid cross, okay, and then next about the gene interaction. In gene interaction, two basic things we saw. One about allelic, another one non-allelic gene interaction. So from that, one after the other, we are going to see. First, we will speak about dominance, incomplete dominance, right? From the word, okay, what can you understand? What is incomplete? Something is not completed, right? The dominance not completed. So it is incomplete dominance. So if the dominance not completed, it will not express its character. So some new character we are going to get that we are going to see in incomplete dominance. I'll show you some example. The first one you can see snapdragon. Another one, a fowl, Andalusian fowl. Here, I'm going to use the word dominant and recessive, okay, but actually they are not going to be a dominant. So you can see white color, okay, as usual, white recessive we can take, and then this pink color, okay, it is a dominant we can write, and a new combination you will get. The same way, Andalusian fowl, if you take it, this is recessive, this is dominant, but you will get a new combination like ash color. Genetically, we call it as bluish ash color. It is so the so-called dominant, but it is not a dominant. The character is not expressed in the F1 generation. So it's lacking. That is why you call it as incomplete dominance, right? So our example, our well-known example, what is there in our textbook, we'll see. It is nothing but Mirabilis jalapa. Mirabilis jalapa, otherwise called as 4 o'clock plant. Okay. You can see the flower color, red color, another one pink color, another one white color. Okay, I'll show you the pink color also. You can see here the combination. So parental type, these two are all parental type. Okay, we'll keep this as a male and this as a female. When these two are intercrossed, you'll get a new combination. It's nothing but what color it is? Pink color. Okay, the same if you want to write the genotype of this, we can write capital R, and here small r, and the pink is nothing but capital R, small r, right? You are getting the genotype capital R and small r, but what color you are getting? You are getting a different color, a blending inheritance, combination of both the color. Pink, that is red color and white color, you get a combination of pink color. Now I'll show you another picture. You can see variety of colors, right? Variety of colors, that is yellow, then red, white, so many types of colors, all in same plant. So there are more variation in this particular plant, Mirabilis jalapa. So we will do a cross, incomplete dominance, like monohybrid cross, but here you will get a different type of ratio. Why I'm using monohybrid? Because one contrasting character we are taking, right? So parental, I'm going to take capital R, small r. What is capital R, capital R? It is red. What is small r? It is white. I'm writing both phenotype as well as genotype. So when it is intercrossed, okay, let me pull out the gametes and then we'll intercross. Capital R, encircle the gamete, capital R. Then small r, small r. And right here, it's gametes. Now, we are going to show the offspring. It is F1 generation. In this, capital R, small r. Okay, the same you will get here. Capital R, small r. So what color it is? On the whole, it is pink color. Okay. Now what I'm going to do, I'm going to self this both the hybrid. On selfing this, okay, what we are going to get. For this, I'm going to get help of Punnett square. Okay, we'll use Punnett square for F2 generation so that all the 4 offsprings I can show. A tabular column, okay, and then divide as male and female here, right here. Female. Okay, let me change the color. Capital R, small r, capital R, small r. I'm taking these two, this one and the other one. Okay, and now here represent with the combination capital R, capital R, capital R, small r, capital R, small r, small r, small r. Now, what are the phenotypes of this particular character? That also will write it, phenotype. So, this phenotype is both are dominant. So, it will show the color red. It is a heterozygous as we are writing here. Okay. So, it should be a pink color. This is also heterozygous. So, it should be a pink color. And then smaller recessive, it is white color. Okay, again I'm using the term recessive dominant means here it is, since we are using the capital letter capital R, small r, because of that we are using. But in the pattern, if you see F1 generation, you will get a combination of that particular 2 colors. So now we'll write the ratios, phenotypic ratio and genotypic ratio. So phenotype, the physical appearance. Okay, phenotypic ratio. What are the colors? Red color. How many red color? And then S2. How many pink color? S2. White color. How many white color? Okay. So how many red? One red. How many pink? Two pink. And how many white? One white color. So we are getting a new thing. 1:2, 2:1. Okay. And then here genotypic ratio. Genotypic ratio. We'll write the alleles. Capital R S2, capital R S2, small r. How many number? Same we are getting. 1:2, 2:1. Just check here. Capital R, small r. Yeah, 1:2, 2:1. The same way the color red, pink, and white. So we get the same number in genotypic ratio as well as phenotypic ratio. It's very important 3-mark question. This cross, they may ask examples for 1-mark question and the cross for 3-mark question. Right? We'll move to the next one. So next is nothing but codominance. What is codominance? We saw incomplete dominance and now we are going to see co. Co is nothing but, that is one over the other, the cofactor we call it as. One color will be dominant over the other color and both will be expressed in the generation, F1 generation or F2 generation. That is called codominance. So we'll see that in still more detail. The alleles which are able to express themselves independently. Okay, red, for example, 2 colors will take it, red color, white color. Okay, both are dominant over each other and the both will be expressed in the next generation. So they are not dependent on the other color. They both show their traits. Even when present together are called codominant alleles. Okay, they show independently even though when they present together it is called codominant, and that phenomenon you call it as codominance. I'll show you an example. Check, actually it is brown. Okay, in genetical way I told you the colors, we call it as red cow, red, white, okay, when it is intercrossed, you will see both the color, white color also, I mean the brown red color also. Yes. And Jersey cow, if you see, okay, black as well as white, a patched work. So both the color are dominant. That is why we call it as codominant alleles. Both the things are expressed. And now, next, our blood group. In our blood group, two things we are going to see. One about this codominance, another one about multiple alleles. Multiple alleles, little later we'll see. Now, codominance. Why should I take this blood group as an example for codominance? Something is dominant over each other. We'll check it out what it is. It is nothing but our AB. In 10th standard, we read about this blood group, right? In genetics lesson. The same thing we are reading, but with a different example here. Our blood group pattern is ABO blood group. And what are the blood groups we are having? A, B, AB, and O. Okay, four types, phenotypes we can say. In that four phenotypes, what will be the genotype? Here we have six genotypes are there. So, for one alone, I mean A blood group alone, two genotypes are there. And for B blood group, again two genotypes are there, right? And ABO only one genotype, O blood group only one genotype. You just observe this one, it is small letter, so it indicates it is a recessive trait. And the capital letter indicates dominant trait. Why I have written A also capital, B also capital? Because both are dominant. A, B, okay, they are dominant over each other. So these two are codominant alleles, whereas I is a recessive allele, right? So this ABO blood group. Still more, what is it? A, B, AB. Why the name has to be given? We'll check it out. So here, this is RBC cell. Okay, in RBC cell, okay, you can see here it is nothing but on the surface there is a globule-like structure. We call it as antigen. So based on the antigen location only, we are saying it is A or B or AB. Suppose there are no antigen, okay, if you see the RBC surface is empty, no such antigen. I'll use one more color for B here, okay, two color. Let me complete and then I'll come to the next one. So this is A antigen, this is B antigen, the black one A antigen, blue one B antigen. If both the antigens are present, it is AB. Suppose here only this blue is present, so what antigen it is? B antigen, so it is a B blood group, it is AB blood group. And here only black color, okay, I'll use the same color so you can easily differentiate. It is A, so it is A blood group. Now here if you see on the surface of this RBC cell, it is RBC, red blood corpuscles. No antigens are there, it is nil, zero. That is why we are given a blood group called what? O. O blood group. Okay, O represents nothing, nil, zero. There are no such antigen. So it is called O blood group, right? We'll see the picture. So it's a O blood group, no antigen. And here you can see one antigen, it's B antigen, so B blood group. And here you can see two antigens, A, B, so it is AB blood group. And here you see only one antigen and that is A antigen, so it is A blood group, right? And now based on this, we can tabulate and say, if these are all the antigen, what type of antibody they will have? The antibody will be the opposite of it. If suppose I am having A blood group and my RBC cell will have A antigen, what antibody I will secrete? I will secrete the opposite. In my blood plasma, I will have the opposite of that, anti-B. Okay. So we'll see here the tabular column. Yes. So this is, I said, no, for example, if I'm A blood group, I will have antigen A, we all know that, and antibody B, anti-B we are having. Why not the same antibody will have if suppose there is same antibody? Okay, imagine this is the antibody A, and my antigen is also A. Okay, this is antigen, my antigen also A. So what happened? They bind each other and they clumps. It leads into precipitation. Okay, we call it technically blood coagulation, clotting. It leads into death also. Okay, that is why naturally we have the opposite antibody, and this antibody is very important. Okay, which gives immunity also. Suppose in blood transfusion, if there is wrong transfusion, A blood group person going to receive B blood group, it leads into blood coagulation. Okay, so that is why every time they will check for the blood group and then only blood transfusion takes place. So similarly, you can see for the B, the opposite anti-A, and A, B, both the antigens are there, so there will be no antibody. And if you see O blood group, there are no antigen, so both the antibodies will be there. Okay. Multiple alleles. Okay, I told you blood group when you take two examples we'll be saying. One is about the codominance A, B. A is dominant over B. And next we are going to see multiple alleles. So whenever we are learning a term, we are describing the particular term and then we are going into the topic, right? So what is mean by multiple? Many. Alleles, okay, the form, the gene form will be more, right? So, what are they? For example, skin color, if you take it, we have many things, okay? Many color combination. For example, coat color of rabbit. Rabbit coat color. Coat means that fur color, if you take it, it has white, it has gray, it has brown, and then it has what combination? The ear alone will be black in color, okay, and then tip of the nose will be black in color, remaining the whole body will be white in color. So different combination will be there. I'll show you the picture. Okay, see here. This is the coat color of rabbit. I said no, a nice combination, right? See, yes. So first, let us understand what is multiple allele. The state of having more than two alternative contrasting characters, more than two alternative contrasting character. Actually, only two contrasting characters so far we read. In Pisum sativum, tall, dwarf, round, wrinkled. Like that, two contrasting characters we read. But here, more than two contrasting character. What color I said? Here they are given no chinchilla, the colored one. Chinchilla, light gray, dark gray, albino, Himalayan albino. Okay, see, these are all the different alleles. So more than two alternative contrasting character they have, which are controlled by multiple alleles. So each one you will give. For example, C, capital C color, capital C, CH, chinchilla, okay, and then albino, C, A, like this. Different alleles you will give. That is why it is called as multiple alleles. Okay, but everything located in the same genetic loci. Okay, loci is nothing but the particular point. So the same multiple alleles, how they are expressing their genotypes. Here you can see. Yes, this is a genotype. And this is a Punnett square. Okay, to find the combination. If two different parents are there, two different color, what will be the genotype of the offspring? Okay, ABO blood group. In next class, we'll see about ABO blood group. Okay, in that, what is the multiple alleles role with some numericals, okay, some solving genetical problems. We'll see. Thank you.